the next section, soil health and organic matter, carbon in the soil. So if we think of soil as being somewhat like a three-legged stool, where the legs are the physical soil, the chemical soil, and the biological soil. Soil organic matter interacts with all three. And if you take one of them away, if you take one of them away, then the stool falls over. So, soil biology fits in with the carbon cycle overall. Carbon, as we know, gets cycled through atmosphere, plants, animals, humans, and the soil. There's about 1,500 gigatons of biological carbon, soil carbon, in the soil worldwide, compared to 750 gigatons of carbon in the atmosphere, or 610 gigatons stored in biomass as, wood or plants or leaves or whatever. And then you've got even more carbon that's stored in the soil as carbonates. So looking down a bit more with a sort of telescope and whatever, within the soil, the carbon's in the biomass, that is the living plants and animals in the soil, and as substrate. So this is dead organic matter, cellulose, leaves, other compounds in the soil. Some of that soil organic carbon is broken down really quickly. This is the active stuff. And there's other kinds of carbon that stay in the soil for years. So if you took a handful of sugar and sprinkled it over your garden, it wouldn't last very long at all. Like, it would be just absorbed into the soil and used by the biology very quickly. Whereas if you go out and put a bunch of wood chips on, they can last for years because it's in a form that's not very easy to break down. So soil carbon on your farm or in your garden. Why should people increase soil carbon? Soil carbon improves the physical condition of the soil, allows better water infiltration. So this goes back to the people having problems with waterlogging. It helps to store water and improves root growth. It's a supply of nutrients. It's a food supply for soil biology, which release the nutrients to plants. And for large-scale projects, building soil carbon over the long term, we're starting to find people getting interested in financial benefits for things like carbon credits or the ability to certify as carbon neutral. So growing your own carbon is what any farmer or gardener already does. The trick is keeping it in the soil once you've got it there. Soil biology uses that carbon as food. It's an energy source. And just like us, they take in carbon in the form of cellulose or sugar or whatever, and they burn and release carbon dioxide back into the atmosphere. So the rate of carbon going in needs to be faster than they're using it for it to build up. In agricultural soils, there's a basic capacity of your soil to hold carbon, and that's determined by the clay content and the climate, basically. So the more clay, the more carbon the soil can store. The more gentle, more beneficial your climate, the higher the rainfall, the gentler the temperature, the more plants you can grow and the more carbon you can put in. So, of course, a sandy desert soil doesn't have a lot of capacity to store carbon, whereas a deep, loamy soil down here in the Fleurieu has got quite a good capacity. Within that, management affects how much carbon the soil can store. So, increasing carbon in agricultural soils. Method one is grow plants. They take carbon out of the air, they leave it in the ground. The amount that stays there over time depends a bit on the system. If you're growing, say, a permanent pasture all year round, so there's green plants always growing, always putting carbon into the soil, that's going to store more carbon than if you, say, growing a crop for a few months, harvesting it and then taking it away. Some of the carbon dioxide that a plant fixes through photosynthesis, it actually exudes out through its roots, and it trades that carbon in sugars, glomalin, sticky stuff, plant slimes and so forth. It trades that to bacteria and fungi in exchange for nutrients. So there's a currency trading market going on underground where the currency is basically carbon. So the plant is trading carbon as energy in exchange for things like nitrogen, phosphorus, sulfur, even water in some cases. And that carbon that goes out through the root system is known as the liquid carbon pathway. The other way carbon gets into the soil from plants is just by the plant's life cycle, dropping leaves on the surface, plants dying, decaying, and so forth. The other way to increase carbon is by bringing it in from other sources. This is like when you're bringing in your compost or your manures or soil additives and so forth. It can be expensive if you need a lot of it, unless there's a really good source that's cheap and close by. Carbon from these sources has nutrients already in it. And, the amount will vary depending on what it is. Things like chicken manure, for example, are quite concentrated, whereas things like sheep manure, less so. This can help to maintain a circular economy, so you're putting nutrients back in where they've been fed out, and it helps to replace those nutrients that have gone. I'll skip past this question unless somebody's got anything urgent, no? Because we're now getting into the basic soil biology, more details. So the soil's a bit of an ecosystem, like a forest or the Great Barrier Reef. You've got a lot of different plants, animals, fungi, bacteria. You've got big things, small things, microscopic things. And they all feed on organic matter from a variety of sources. There's a lot of different organisms adapted to different soil conditions. So some bacteria, for example, really like soils that are, say, high in phosphorus or high in nitrogen. Others live in low nutrient soils. Earthworms tend to like to live in soils that have moisture or enough moisture through the year. If they dry out, the earthworms either go deep in the soil to where the moisture is, or they have to hibernate. Termites and ants, on the other hand, can survive in a wide range of conditions, and they do really well in drier areas. They don't need the moisture that an earthworm does. These organisms are all affected by what happens to the soil. If you dig it up, you're digging up this soil biology. If you add nutrients to it, you're giving food to soil biology. If you put too much on, you're drowning the poor beggars. So they're using those plant and animal materials. They help to create soil humus. They recycle carbon, nitrogen and other nutrients, and they support plant growth. And they can do this directly. There are some soil biological organisms that will directly interact with plants, and they can be either beneficial, neutral or harmful. We all know that there are biological things out there, fungi, nematodes or so forth, that are parasites. They affect plants badly. There are other ones out there that affect plants in a good way. So having a close-up look at some of these things, bacteria under the microscope, very important. You've got your teaspoon of soil there, for example. There could be 5 billion bacteria in that soil, as long as you're feeding them and keeping them alive. There's thousands of different species and they've got a lot of different functions. Mostly bacteria mobilise nutrients and circulate them, and they may be associated with plants like the nitrogen-fixing rhizobia that live with legumes. Fungi, also extremely important. Could be as many as 20,000 kilometres of network. So if you strung it all out one fungi filament long, you'd have 20,000 kilometres in a cubic metre of soil. Fungi can physically bind the soil together. they can decompose organic material, and they can have symbiotic relationships with plants. This picture here is a pine tree. You can see its main root structure in the middle, and around the outside are the mycorrhizal fungi that are symbiotic with that plant. If you ever go into the pine forests in the Adelaide Hills at the right time of year, you'll see little mushrooms called pine mushrooms or saffron mushrooms, and they're a sort of light brown colour with light dapples on them, milk spots. They were deliberately introduced to the pine forests because they are symbiotic with pine trees and they improve the growth of the trees. Amoeba, up to 5 million per teaspoon of soil. These help to recycle nutrients by feeding on the bacteria. Breaking them down, their remains go into the soil and get taken up by plants and fungi. Nematodes, believe it or not, this is actually a friendly beast. The picture there is a nematode that eats other nematodes and helps to keep the nematode population under control. There's a lot of different species. Some will attack crops and pastures. If you ever grow tomatoes, you don't want to grow tomatoes 2 years in a row on the same soil because there are nematodes that increase in the soil in the first year and they'll overwhelm your tomato plants in the second year. Could have as many as 5,000 per teaspoon. And again, they're nutrient recyclers. By feeding on fungi and bacteria and plant roots and other things, they help to cycle nutrients through the soil. So getting into things that we can see with the naked eye now, earthworms, they decompose smaller organisms when they eat through the soil. They also take in things like leaves, roots and so forth, take them in, digest them and break them down. Their excretions help to bind the soil. They actually have an amazing variety of antibiotics in their gut, which I suppose they need because they eat bacteria and so forth and they don't want to be poisoned by them. And vermicompost, that is the compost that's been through earthworms, is very rich in plant growth promoting products. They're an indicator species for soil health in the higher rainfall areas. Basically, the more earthworms you have shows the more healthy your soil is. Being earthworms travelling through the soil, if you have few ones, it is probably either because the soil is very dry or very compact because they have difficulty getting through and therefore getting enough to eat in a compacted soil, or it may be disturbed on a regular basis because when earthworms are dug up, it can physically damage them. It can interrupt their breeding cycle and damage eggs. And in general, it's like your home is getting continuously destroyed and you have to rebuild it. If they're sort of, allowed to live their lives in a secure house, they're more likely to be able to spread and reproduce. Other things, spiders, mollusks, those things with shells, snails, beetles, ants, termites, etc. They burrow, they turn over the soil, they mobilise nutrients. Ants come up to the surface and grab stuff and bury it. They break down the organic matter. Larger species, mice and rabbits and so forth, also loosen the soil, dig things. Can't say I'm particularly fond of mice because they get into my pantry, but as long as they stay out in the soil, all good. Interestingly, some of these macrofauna, such as termites, they help to add nitrogen to the soil because they have bacteria in their gut that fix nitrogen from the air. So the termite itself isn't doing anything, but it's a host for bacteria which are helping the soil. One of the reasons that keeping stubbles in the paddock and not burning them on everywhere is that it's provided a lot more extra food for termites, which are then able to build up numbers and more nitrogen goes into the soil. It it just takes a few years for them to build up. So termites in garden or paddock, good. Termites in house, bad. And they're probably completely different species. Soil biology, most of it's found in the topsoil, that top 10 centimetres, because that's where their main food supply is. A lot of their food supply comes in from stuff that lands on the surface. The biological activity also needs moisture, the right temperatures, and oxygen. When the conditions become unfavourable, these organisms can either die or shut down their activity until the conditions are right again. So this is things like earthworms going down to deep conditions and hibernating. In the case of fungi, the fungi may die out, leaving spores in the soil, which would germinate and grow when moisture comes back, for example. However, long-term changes to the soil environment will change the balance of that soil ecology. Now, sometimes this can be in a good way. For instance, if somebody has been in a cropping situation and they change from ploughing every year to no-till, that's a change to the soil system that will be beneficial, but the soil biology has to find a new balance within that. If somebody changes from dryland production to irrigated, then the soil biology is going to have to find a new balance. Some things will, obviously grow like mad because now they've got moisture all year round instead of just in winter. So the cycling of the soil organic matter by the bacteria and the fungi and the other organisms allows the nutrients to be made available to be taken up by plants. If you're applying a synthetic fertiliser, no matter how water soluble it says it is, 80% of nitrogen fertilisers are cycled by soil biology before they're taken up by plants. It's a similar figure with phosphorus. Having functional soil biology in the soil is very, very important. So, and also storage and release of nutrients from organic matter through the soil is an important part of that nutrition. Any organic matter that's applied to the soil pretty much has to be cycled by soil biology before it's available to plants. This is why they say, put on your compost and organic fertilisers a few weeks before flowering and so forth. It gives the soil biology time to start breaking down and cycling and making it available to the plant so it's there at the right time. So each farming system or garden or whatever has its own characteristics which can favour various kinds of soil organisms or be unfavourable to others. See, personally, I'm not a big fan of cockroaches in the garden. I know they break them down, but my garden, I squish them when I see them, and the same thing with snails. So you could say that my garden is unfavourable to snails just because of me. A different system with a different gardener might be perfectly fine for snails and cockroaches. The type of farming system is going to determine the mix of the species present. So this is things like rotations, different kinds of plants and so forth, whether chemicals are used or not used. So any system which promotes a good balance of beneficial soil organisms is likely to have fertile soil, productive soil, well-structured and resilient to adverse conditions like drought, resulting in healthy crops and pastures. A few notes about practices in systems. Excessive tillage breaks up the soil structure and the fungal hyphae networks. So your 20,000 kilometres gets broken up into little bits of 1 kilometre each or something, and it doesn't work as well. So that reduces fungal activity and it lowers the population of things like earthworms. Tillage can also alter the oxygen availability in the soil. What you get is a flush of bacterial activity initially, which makes nutrients available quickly. This is why people used to plough before they seeded. You get this flush of nutrition and the plants can take off. But it reduces it in the long term because it's all used up at the start. Soil pH affects soil biology. The bacteria and earthworms won't thrive in acidic conditions. When you consider that a soil with a pH of 4 is about as acidic as lemon juice, you can understand why an earthworm is not going to survive in those conditions. Pesticide and herbicide applications usually only have a short-term effect unless they're used repeatedly over a long time. Effects vary depending on the chemical and the soil type, and of course a fumigant will kill all the organisms and it will take time for them to come back. So with soil biology and farming or garden systems, the amount of activity in the soil depends on the inputs available. So your biology needs your food, water, air and shelter. Food is mostly organic matter. It also supplies shelter. So a soil that's low in organic matter will have low biological activity. A lot of Australian soils around the country have had organic matter levels reduced through past farming activities like burning and bare fallows. As those practices have basically disappeared from the landscape on the whole, the soil organic matter levels are rising again. I mean, like, it's been tracked over time that as things like no-till and stubble retention have come in, soil organic matter levels have started to go up again, which is great. So if you want to increase biology in the soil, basically increase the food supply, because you're not going to be able to do much about rainfall or temperature unless you're applying irrigation and heating systems, which is not likely. So keeping green plants growing as long as possible, reducing tillage, retaining stubbles or organic matter on the surface, which is linked to increasing ground cover. Mulching is also a form of ground cover that can be used, particularly under vines, trees or horticultural crops. And other inputs with carbon content can be useful when they're used correctly. So in summary, remember that your soil biology is affected by your soil chemistry and your physical properties. So get the chemistry and the structure of the soil right, or they won't survive, or if there's no food there, then they can't survive. So there's not much point adding new soil organisms if they can't survive in the soil. On the other hand, get the conditions right and the soil organisms will thrive and multiply. So have we got any questions on soil biology or soil carbon for that matter? Not online?
If the soil is dry, is there a way to rectify?
So the question is, if the soil's dry, is there a way to wet it again? Now, is this about the water repellents or just dry soil?
Yeah, for example, if you went away for 6 weeks and came back, it's all dried out.
Yeah. Okay. So if you went away and your garden was all dry out, can you get it back to what it was? Yes, it can. The soil biology is resilient enough to take that kind of thing, and it's a natural process in this part of the world, in that the soil does dry out and get quite warm over summer, and then gets wet again in autumn. So if it's a regular kind of cycle, then yes, the soil biology will be able to handle it. If you've got a very delicate plant and it's died, well, that's just the way it goes.
With the microbiological mixes that you can get at like garden stores, like a Cherry Forte or something, is that just a broad spectrum of bacteria and soil microbes that they're adding?
Yeah, so the question is about the biological mixes that you can get in stores to add to the soil, and are they just a broad spectrum or are they specific? That's going to depend on the product. Most of them are probably a broad spectrum. They will they're likely to have some nitrogen fixes in there because getting a bit of nitrogen into the soil will make your plants look better and you'll be able to see that there's something happening. You can buy specific things if you have a need for it. So, for example, farmers that grow legumes can buy inoculants with rhizobia so that they have the right soil biology right next to the seed when the plant starts growing. In horticultural situations, you can get specific things so that you can buy, for instance, beneficial nematodes to go in and clean out all the parasitic nematodes. You can buy specific fungi that interact with soil roots in a good way and crowd out parasitic fungi. So the generic ones are probably a mix, but you can get specific ones. They are on the market. There's another question.
It's been a tap water.
Tap water.
Yeah, what's the effect on the biology?
Your average tap water, it's got a little bit of chlorine in it, but it is low enough that if you've been using tap water on your garden, it is probably having no effect whatsoever apart from providing water, because the biology in your garden, if it's been watered for a while, they are completely used to whatever's in that tap water and it's already adapted. If you were going from a never watered situation to now being watered with mains water, the benefits to the biology are going to far outweigh any effects of the additives in the tap water. So I would say overall, beneficial. It is theoretically possible to get chloride toxicity, but it really only occurs in places like right next to your swimming pool where you've got a really high chlorine concentration and it splashes over and you might get a little bit of an effect right on the edge of the lawn. That's it. I believe that we are at 8:15. I can tell you a little bit more about testing for soil carbon if you're interested. I've got a few notes. All right, we'll go on to soil carbon. Okay, so if you're testing for soil carbon, you want to know what's going on with the carbon in your soil. There's two main tests that you can get for soil carbon at the moment. One is the total soil carbon. It's also known as the LECO test because LECO is the machine that's used. The second one is the Walkley-Black soil carbon test. Total soil carbon measures all the carbon in the soil from any source, and that includes from rocks or minerals like lime. Now, South Australian soils have got a variety of soils, and outside of the Adelaide Hills and the Fleurieu, quite a lot of them have got a lot of carbonate in them. In this particular area, probably you don't have carbonate unless you're next to Waipinga or some of the areas where they're mining lime. So if there's carbonate present, it needs an acid wash to remove it before they do the LECO test. That's the only difference it makes. The LECO test is relatively recent. It came up in the last 10 years or so. Before that, they used Walkley-Black organic carbon. That measures carbon from the soil, but not from rocks or minerals. So it won't measure the carbonates. It's a widely used test and it's still used in areas where any carbonates are present, but it can slightly underestimate carbon levels compared to total soil carbon. So if you order a soil test, those are the two kinds of carbon that may be used by the lab. Total soil carbon is now the standard and it's cheaper, but some people, if they've got records of their soil carbon over many years, will still use Walkley-Black because that's what they've got going back 20 or 30 years and they want to maintain that continuity. skip to soil biological activity and testing. So soil biology is a pretty recent science. If you want to get an animal named after you, find a soil biological animal and you can get it named after you, because there's heaps out there that haven't been discovered yet. There's a lot of different tests available, and there's a number of different labs, and they're all the best. They say so themselves. But broadly speaking, there's three kinds of soil biology tests. There's population analysis, activity analysis, and indirect indicators. So population analysis tells you how many there are of your chosen organism at a specific time and place. So this is things like counting earthworms, sending a soil sample to the lab to have bacterial counts done, etc. They are specific conditions at a specific time, and particularly biology or fungi, they'll change throughout the year. So you might sample in spring and go, whoa, I've got heaps of soil biology, look at that. You sample 3 months later, everything's dried out, and it's like the numbers have completely disappeared. So if you want to track things over time, year on year, you need to, as much as possible, sample at the same time and same conditions in each year. They are useful in that things like farming practices can very quickly affect these populations. So if you want to know what difference, say, putting on a biological additive has done, you can go out there, put on your additive, leave an area so that you've got something to compare against, and then you can take a sample from the control and a sample from your treated area and have a look at the difference. And you can also do that over time, at say a monthly interval or whatever, to see how long an effect might last. But it's always good to have the control there, because you might find that your effect is decreasing over time. But is that because the numbers are actually disappearing because of something you've done, or is it that the numbers are disappearing because the soil is drying out? This is why having something to compare it against is useful. You can get general tests and say, it'll measure all the bacteria in your soil or all the fungi, or you can get specific tests. So if you want to know whether there's an organism in your soil which is going to affect your next lettuce crop or something, some kind of wilt, you can test for just that one, which you know is a pest. Other measurements, you can do things like nematode counts. Is it a good idea to plant this particular crop which is susceptible to nematodes, or are the numbers too high and I should plant something that's resistant to nematodes, that kind of thing. So activity tests measure what the soil population's doing. They are pretty sensitive to environmental management, similar to the population. So examples are carbon dioxide respiration measurements. You can take a sample of soil and you wet it up so that it's active, and put it in a little bowl and stick the little indicator strip in there and put the lid on and come back the next day, and if it's turned purple, you know that there's some carbon dioxide in there. You can get kits for these tests so you can see how much activity is in the soil. There's the cotton strip or the underpants test, and that measures the breakdown of cellulose. That is, like cotton is cellulose, so you take a piece of cotton, bury it in the soil and leave it there for a certain number of weeks, mark the spot where you buried it, because you're going to come back, dig it up and see how much is left. And in some cases, like if you bury a piece of cotton in a dry, sandy soil, you can come back 6 weeks later and it's practically brand new. There might be a couple of brown marks on it, and if it was the underpants, you hope that that was the soil. Whereas if you take your piece of cotton and bury it underneath an actively growing, say, garden bed that's got, lots of activities, spiders, earthworms or whatever, you might have to come back in 2 weeks because there are things there that break down cellulose really quickly. And you could come back and you might find that you've got a thin little bit of cotton and there's a lot of holes in it and there's earthworms all over it, chewing away and trying to get the last little bits. Or in the case of underpants, you come out and they go, there's just the elastic left. Oh well, great biologically active soil. I had a lemon tree where I'd been dumping the lawn clippings for several years. And at first the lawn clippings would stay there for months. They'd just sit there. But after a while, the biological activity in the soil got used to the idea that they were getting regularly fed. And I would put lawn clippings down on that spot and they'd be gone in a couple of weeks. And the soil in that spot was dark brown, rich and crumbly compared to the lawn, which, because I was living in Salisbury at the time, was a pretty sticky red clay. And you can get other more specific soil biological tests. So some farmers will, for instance, go out and find out how quickly is my soil producing nitrogen for plants? How quickly is it cycling phosphorus? Have I got enough activity in the soil to supply my plants, or do I need to add some extra or boost the soil activity so that my plants get the nutrients they need as quickly as they need it? And then we have indirect indicators. So these are generally indicators of soil health, like your organic carbon content, so going back to your total carbon test or your Walkley Black, or observation things like ground cover. For example, if your organic carbon content is very low or your ground cover's just not there, there's probably a problem present and it's going to be affecting your soil biology. On the other hand, if your ground cover has stacked up and it's not getting broken down at all, you've got a problem there in that your soil biology is not cycling effects. You're providing it with food, but your biology is not able to cope with it. So it might need a bit of help. If you provide them too much carbon, but not enough nitrogen, then they'll be limited in what they can break down in the carbon because they're starving for a bit of nitrogen in the system. So again, if you're testing, ask yourself, what do you want to find out? Why do you need to know that? And how's the test going to help you make management decisions? Sometimes it's, I'm just curious. I want to know what's going on in my soil, how much bacteria, how much fungi or whatever. In other cases, particularly for commercial or semi-commercial growers, it's like there's something going on here. I know my nutrients and structure are good. There must be something happening with the soil biology that I need to know about. And this is cases where, for example, the ginger growers in Queensland had real problems where they'd grown ginger year on year and suddenly their crops were failing. Turned out there was a root rot disease that had come in, and because the ginger was getting grown there year on year, it had finally built up to the point where the crops would fail. And in that case, the solution was rotate your crops a bit so that you're not hosting that disease and letting it build up. Another thing that's good to do with biological testing is find out the requirements of the lab that you're sending them to. There's no point sending a sample if it's not going to be any use when they get it there. So how much do they need? Some labs will say we need 500 grams. Some labs will say we need 2 kilos. Some people will say we need 100 grams. Postage costs money. Find out how much you need. Doesn't need any special treatment. Some labs will say you need to get this soil to us within 48 hours and it has to be kept refrigerated. That's important. Other labs will say, no, it's okay. However, leaving your sample in a plastic bag on the dashboard of your car for a week in the sun, not a good idea. We did biological testing with 4 different labs once to compare the results between them. one of the labs had a standard procedure when the sample came in. They did not read the label. They chucked it all in the drying oven. That particular lab had way lower biological activity than all the others because they just killed it. In that case, it wasn't our fault. We collected it correctly. They processed it wrong over there. That lab doesn't do soil biology tests anymore. Now, do they have any specific sampling requirements? Does it, if you're in a vineyard, do you have to do it under the vine or in the middle of the row? If you've got horticulture, do you have to do it like on a mound or in between? Does the soil have to be moist? Standard nutrient testing is done in summer and autumn when the soil is dry because the soil is stable then. It's not cycling the nutrients usually. But biological testing is quite often done in spring when the soil is moist and things are cycling and soil biological activity is at its highest. So if the lab has a requirement for a time of year, then it's good to know about that. A few more tips. Because soil biology is so sensitive to environmental conditions, monitoring it over time gives a better, more complete picture than just a one-off test. However, if you're looking for a disease or a problem, a one-off test may be all you need. Comparing tests should be done with using samples that are as similar as possible. If you've got two separate soil types in the same paddock, don't mix them, otherwise you'll get something that is actually not representing either of them. Even samples from the same soil are going to vary through the year, depending on moisture, temperature and food availability. If you want a typical test, try to avoid sampling just after doing something like spraying or seeding. And use a reputable laboratory, like one that's been around for a while and has a good reputation. Because it's a new science, stuff comes up and you sort of get pop-up labs. Use the ones that have been there for a while and are well thought of. And if you're after more information on soil carbon and soil biology, there's some useful fact sheets out there. Caring for Our Country, CFOC, and the Wheatbelt NRM has got a website called thesoilquality.org.au, and that has some interesting information on soils. It's geared towards cropping and pastures, but the principles are the same no matter what. And there are other sites based in Victorian DPI and the Queensland DAF Healthy Soils. So I hope that might be useful to you. If there's any questions, now's the time. And yeah, while you're thinking of that, I'll just acknowledge the Willunga Environment Centre for hosting today. Thank you very much. And Janine and friends for organising and making it possible to do this online as well as in person. That's great. The Hills and Fleurieu Landscape Board and funding from the Australian Government's National Landcare Program.
I have a question. If people have further questions and they actually do want to get some assistance on their properties with their soil, who would they contact? Would they come to the landscapes office down the road, or is there somewhere else that they might go?
Okay, the question is if people want assistance with their soils, who can they ask, and is it the Landscape Board? Yes, we can help with soil problems. We're really big on helping to prevent erosion and that kind of thing, and improving soil health, keeping cover on soils and that kind of thing. We can offer general advice about nutrition and soil health in that respect. It's probably easiest in the current COVID circumstances to send an email either to the Hills and Fleurieu Landscape Board Inquiries, which is on our website, or to call, and if you call the office, you will be put onto the most appropriate person to help you with your inquiry. And the landscape board also assists with pest, plants and animals, things like sampling. We can lend out soil samplers. We can have people go to your property to talk about land management and property planning as well. So that may be useful.
Thanks. Questions? Okay. Well, on that note, thank you very much, Rebecca. That was really informative. Well, actually, written quite a few notes. It was really good for me. So if we just thank Rebecca for coming out and giving this talk, that would be great. My friend here is going to help me put this all together somehow and make a YouTube. So this will be available online at some point, which will be great for the people at home as well, because they would have missed a bit of the middle bits where we were doing some testing. So, and then we'll be able to share that as well.
Yes, and Janine does have the PowerPoint, and I'll send you the PDF of the PowerPoint so that you can email that out to everybody that would like a copy.
Okay, excellent. All right. Thanks very much, everybody, for coming out.